Hello, I'm Dr. Robert Sizer of Roosevelt University, and in this video I'll be talking about immunity and vaccine action. This is the first video in our third module in our series on online vaccination resources. In this first video, first part of the module, I've titled it From Naive to Immune, and I'll be looking at the following topics. First, the importance of awareness in our interactions, scaling from the community level down to the cellular level, the cells and molecules of the immune system, innate and adaptive immunity, and the definitions and explanations for how that works in our bodies, protective immunity and memory, and finally, an introduction to vaccines and the process of immunization. I'll close with a look at herd immunity, which is a term that you certainly will have heard about in various descriptions of our response to the COVID-19 pandemic. So the first thing I want to talk about is the importance of awareness in our interactions, and talk about what that means in different contexts. The animation that you're seeing on the right side of your screen shows a simulation of interactions among people when there are distancing guidelines in effect and people are obeying them, and when there are no distancing guidelines in effect. The dots shown in green are healthy individuals, the dots in red are infected individuals, and the dots in yellow are individuals who have been infected and then recovered. First thing you'll notice is that there's a lot of movement on the left side, and you see a lot of colors changing as well. On the right side, you see less movement. Fewer of the individuals are moving around and interacting with others. So the outcome of that, which we're attributing to social distancing and the limitation of movement, means that more people stay healthy. And at any given time, fewer people are infected, and people are more likely to recover from their infection without spreading the disease to somebody else. So the simulation shows how disease spread can depend on person-to-person interactions. This is something that we've all heard about, but and we've all tried to practice, but it's interesting, I think, and illustrative to look at this in animation and think about how our decisions about whether, where, and with whom we interact are directly related to the transmissibility of a disease. The same thing happens, the same principles apply at the cellular level. So in this image, we're seeing the interactions among cells in our body. The image, the part of the cell on the left side of the image is a lung cell that has been infected with the SARS-CoV-2 virus that is responsible for the COVID-19 disease. The other two cells shown in this diagram are cells of the immune system, a T cell and a dendritic cell. And you'll notice that these cells are all interacting with each other. Their ability to recognize each other is a factor in the recognition and control of the infection. So at the cellular level, even as at the individual level, cells interact with each other and they make decisions about what to do in response. And this is the basis for the functioning of our immune system. So if we scale back out now to from the cellular level all the way out to the individual, we can talk about what happens when people are interacting with each other depending on their degree of immunity to a disease. In this diagram, the top part of the diagram shows us the interaction between a person who is infected with a pathogen, we assume it to be a virus by the way it's drawn, and their interaction with someone who is naive, who has not yet mounted any type of immune response, has not yet encountered that pathogen. When the infected person spreads the virus to a naive person, the naive person mounts what's called a primary immune response. Their body undertakes a series of cellular and molecular interactions in order to recognize and try to deal with that infection by that pathogen. As a result of that, the naive person mounts that response and becomes symptomatic. They are likely to get sick in most cases, and they are likely to transmit that pathogen to someone else that they come in contact with. After they have mounted that immune response and have started to develop immunity, if they are then reinfected by someone who has that pathogen and shares it with them, transmits it to them, if they are immune, they can mount a secondary immune response. That secondary immune response means that they can deal with that pathogen more effectively and are likely to not be symptomatic. They are likely to not get sick as a result. Also, they are less likely to transmit that disease, that pathogen, to other people. So the ability to resist disease, the ability to build immunity, is based on these cellular interactions. And in immunological terms, we call this self and non-self recognition. It requires the person who has not yet encountered that pathogen to be able to recognize it as something that is not belonging to that person, not belonging to the individual. It's non-self. And so the immune system is stimulated to fight it. So our infections then are less likely to make us sick if our body knows that we've been infected before, if we already recognize and know what to do with something that we don't see as part of our own body. So here are some of the cells and molecules that are responsible for this, this response. It's a whole coordinated effort, works over different timescales and different levels to help us recognize those cells and those molecules that are ours and those that are not ours. So to distinguish between self and non-self. So this is just a gallery of those different materials. Collectively, their workings are known as the immune system, specialized cells found throughout the body. And its job is to monitor, recognize, and target non-self molecules. And the term that we use to describe the action of this, this system, this collection of cells and molecules and tissues, is immunity. Immunity is the parts of the system working together to fight infections. Now, as I mentioned, we classify immunity into a couple of different categories, just depending on when it happens, how long it lasts, whether it's specific to a particular pathogen like a particular virus or non-specific, etc. So the first distinction I'll make is between adaptive and innate immunity. But the upper panel is a sort of flowchart for innate immunity. This is a quick and non-specific response. It relies on recognitions of pathogens by sensor molecules that are on the surface of select cells. It activates cells and inflammatory molecules that will lead to many of the symptoms that we associate with that first infection: high fever, the chills, histamine responses, things like that. The innate response is not specific to a particular pathogen, but it does tend to lead to the removal of the infectious agent, or at least to a lowering of the amount of that infectious agent or pathogen that's in the body. The adaptive immune response can start within a few days of the innate immune response. So it is over the longer term, and it can last for several days, and it is more specific. So this is the body's mounting of a defense that is tailor-made for that particular pathogen. It involves T cells and B cells, which are major components of the immune system. These cells are effectively selected and trained to work in a specific way against that specific infection. They can migrate to the site of infection, where they are tasked with generating antibodies, killing cells that are infected, and also overall leading to the removal of that infectious agent. So the innate and adaptive responses can both happen in response to infection. Here's a schematic of the, or here's a diagram of the innate response showing us how it might work in practice. In the case of a viral infection, the virus can be internalized and brought into cells where it could be received and recognized by pattern receptors. It can also be recognized on the outside of the cell. In either case, signals are sent to the nucleus of that cell that stimulate it to make specific proteins. Those proteins then can be released from the cell and can serve as signals for other cells to become active. These will recruit cells to come in and to identify the virus and begin to neutralize it as a general threat to the body. And this can happen very rapidly. The adaptive immune response, as I mentioned, takes a little bit longer because it requires a specific response. But that specific response can be much more effective at dealing with that specific pathogen, especially upon reinfection. So in this response, a virus will infect and replicate within the body. It will be recognized and picked up by so-called antigen presenting cells, which will take it over to the lymph nodes, which are specialized tissues in the body where immune responses are mounted. It's in the lymph nodes that the B cells and T cells that I mentioned before are going to be activated to make a specific response. The T cells are going to have molecules on the outside of their their surfaces that are going to be able to recognize the pathogen and be able to come in contact with other cells and stimulate them to take action or stimulate them to self-destruct or to meet with several other different types of fates. The B cells are going to be able to become activated and will make antibodies, which can then be released. So the net effect is to have a specific response to that infection that is mediated by both cellular methods and by so-called humoral methods, meaning the production of antibodies. So altogether then, the infection with a pathogen leads to a coordinated immune response. And an additional benefit of this response is that through the formation and selection of these specific B cells and T cells, the body can form what's called an immunological memory. So it can remember its interactions with that pathogen and be prepared to make those antibodies again and be able to make those cellular responses again in the case of a later infection. And that is, again, the basis of what we know as immunity or protective immunity. So this graph shows us how this immune response can look over time using the production of antibodies as a measure. So again, under initial exposure, the first time your body sees a pathogen, it will mount that primary immune response, and it will use the innate and adaptive arms of the immune response in order to begin to make antibodies and to try out other defenses, all for the purpose of reducing the amount of the pathogen that is in your body and to keep it from spreading. After some time, you build up protective immunity and memory, and that begins to protect you. Even if you don't have a lot of antibodies around, you still have the cells that can produce them. You still have the T cells that can stimulate other types of responses. So upon secondary exposure, and that can be the second, third, fourth, fifth time you encounter that pathogen, your body will be able to mount that secondary immune response and will be able to quickly make the molecules and produce larger numbers of cells that are already trained to mount a response. And that means that you may not even get sick because your body can deal with that infection very rapidly. So let's go back to our diagram of what happens to the person who has not yet encountered a pathogen. As I showed you previously, if they are naive and come in contact with someone who's infected, they may become symptomatic. As their body mounts their response, they can get sick. And in the case of several types of infections, and with COVID-19 as well, not only will people become symptomatic, they may become severely ill and are at risk of death based on a number of different factors. So even as the body is trying to get rid of the pathogen, it can become people can get very, very sick, and of course are at risk of death if their reaction is too strong. Assuming that they survive the initial infection, the formation of the secondary immune response means that they can become immune. They are then, upon reinfection, typically asymptomatic and less likely to spread or fall sick, fall ill from the disease. So this is the basis of protective immunity, meaning we can avoid getting sick if we're exposed to someone who's infected. It also means that we're less likely to infect others. But a major problem of this, of course, is that there are many complications. And getting sick to build immunity is a risky proposition. And this is where vaccines come in. So the idea behind a vaccine is to stimulate the immune response for protective immunity without the risk of contracting disease. So in vaccination, the form of the pathogen, an inactive form of the pathogen, is given to someone who is naive, who is immunologically naive and has not encountered it yet. They mount their primary immune response just as they would in the case of the live pathogen. They may experience mild to moderate symptoms that last for a short period of time, but they don't have the disease. If they are then exposed to someone who is infected with the live pathogen, they can still mount the same secondary immune response, and they can recover and are also less likely to spread the disease. So the immunity is obtained not by interactions with people who are infected with the live virus, but through the administration of a vaccine that stimulates the immune response and leads to the same end, but without the risk of severe disease. So this practice, this idea of being able to sort of prepare someone's immune system for an infection, goes back a long time, hundreds and hundreds of years, and has been experienced in various forms. The first form that we know of is called variolation, and this relied on exposure to small amounts of a pathogen in order to stimulate a milder reaction. In the case of smallpox, as shown in the image here, the practice was to take dried forms of a virus that came from someone who was infected, and to blow that into someone's nose. They would get sick. In many cases, they would recover. And so if they had a more extensive exposure to a higher amount of the pathogen later on, they had already built the immunity. So again, this was in practice in several different parts of the world for hundreds of years. And it predates what we now consider, what we now call vaccination. Vaccination is exposure to a less dangerous version of a pathogen, and it's a safer way of building protective immunity. Among the more famous examples are those experiments carried out by Edward Jenner using cowpox, which is a pathogen that is related to smallpox, the first smallpox being the first disease to be completely eradicated in the world, thanks to approaches like this. So an example of how this works is that someone who is infected with cowpox becomes the source of material in order to vaccinate or inoculate someone. In this case, a young person who was immunologically naive, had not yet encountered smallpox or cowpox, is given cowpox isolated from someone who has been infected with it. They fall ill with a mild case of cowpox, and then the, the, the the proof of the protective effect was shown upon further exposure to someone who had contracted smallpox, and then the person who was inoculated with cowpox did not get sick. So this showed that protection could come from not only the same pathogen, but from a related pathogen that was less dangerous and was less likely to lead to severe disease. So modern vaccines are based on the same principle as the cowpox vaccination against smallpox, but they're significantly improved in the respect that the vaccines that we deliver now are not active and they can't replicate within the body. It's not substituting one pathogen for another. It is using the molecules of a pathogen that will stimulate an immune response without having the risk of actual infection and spread. So that leads me to the final portion, which kind of hopefully brings this all together, and that's to talk about the idea of herd immunity. So herd immunity is protective immunity, and it is an extent of protective immunity among a population that will significantly inhibit the spread of a pathogen and lower the risk of illness and mortality. You'll remember at the beginning that I showed you this animation of what happens with disease spread in the case of social distancing and in the absence of social distancing. So that is shown in the first two rows of this diagram. Again, we have healthy individuals in green, we have infected individuals in red, and we have individuals who have acquired immunity in yellow. And those who are deceased as a result of the disease, people who died from it, are shown in gray. So in the absence of immunization, the absence of social distancing, the uncontrolled spread of disease, a few infected individuals will infect others, and the infection passes freely among individuals, some of whom will recover on their own, some of whom will succumb to disease. So that at the end of the process, everyone has been infected, some have recovered, and some have died. In the presence of social distancing, when actions are taken to moderate and regulate our interactions with each other, people who are socially distancing and are controlling with whom they interact are not exposed to the pathogen. So you can start with the same number of infected individuals, fewer of them will be passing it to others, and fewer people who are healthy will be coming in contact with people who have been infected. So the isolation of individuals slows the spread of infection. Here, everyone who has a box around them is social distancing. So only people who are not staying socially distant and are coming in contact with people who are infected will become infected themselves. So over time, you will have some people who are infected and recover. You will have some people who contract a severe disease and may die, but more than half of the people will remain healthy. In the case of immunization, where people are vaccinated against the disease, against the pathogen, you can start with the same number of infected individuals, And at the beginning, a larger number of people have already developed immunity through vaccination, and a few have not. So the people who are immune serve as a barrier to the spread of the infection, because if they come in contact with someone who is infected, they are less likely to get sick and to spread it to others. So the infection cannot pass freely among individuals. And those who are healthy will tend to stay healthy. Those who are immune will stay immune. And those who are infected may recover without spreading the disease to others. So in the end, you have people who are still immune, you have people who have recovered, and you have a much lower mortality rate because the number of people who are infected is much lower. Notice here that not every single person is shown in yellow at the beginning. And this is what we mean by herd immunity, that we do not need to have every single person in a population vaccinated and have immunity acquired prior to the presence of the pathogen in the population. We can stop the spread of the disease without having every single individual vaccinated and immune. However, we do need a critical number, and the estimate is typically around 70% of the population, so that people who may not be able to receive a vaccine are able to be protected due to the immunity of others in that population. So the ideal situation that we want to have is actually some combination of immunization and continued social distancing, because even with vaccination, it may be possible for someone who is immune to pass along the virus to someone else. So ideally, we want to be able to continue to monitor our interactions with each other, even as we develop and use vaccines as a way of building herd immunity. So the one sentence summary to sum up this first part of the module is that vaccines train your body to fight germs faster and with less risk of illness. In the second part of this module, I will be talking a little bit more about what vaccines are like and how they're developed and produced and approved. So I hope you'll watch the second part along with all the other videos in this series.